Hello, good morning, everyone. Welcome to the virtual online lecture for today in our GeneTime conference. I'm very happy to introduce you Sidonie Bellot from Kew Botanical Gardens. She did the PhD in the Ludwig Maximilian University of Munich, and she's expert on genome evolution, phylogenomics, biogeography, seed trait evolution, barcoding, the DNA barcoding of plants, and some other very interesting topics. And I would like to thank you, Sidonie, for accepting our invitation. And please take your time.
Well, thank you, Fernanda. Thank you for inviting me. Actually, I'm happy to talk to you about this topic today, which is the potential and challenges of DNA barcoding to support plant conservation. So my talk will be divided in 3 parts. So first, I will say why I'm interested in DNA barcoding and why it can help supporting plant conservation. And then I will spend a bit of time showing the different approaches of DNA barcoding, explaining a bit how it works. And finally, I hope to finish with real case studies where we can see a bit the challenges and the potential of this approach. So to start with the why, as you know, many plants are threatened with extinction, and there are many reasons for this extinction to happen. For instance, agriculture and aquaculture and land use change, and but also another main reason is, for instance, the use of species for providing services to humanity, or for instance, destroying ecosystems to get minerals from the earth, etc., etc. So many reasons, and in fact, DNA barcoding might help to tackle many of those problems. As I said, one of the main reasons is species overexploitation. So for instance, here we have a very well-known case, which is the case of rosewood, which is used a lot to make luxury furnitures, for instance, so guitars. And there are 2 problems with this. First, the species that are used themselves, they are decreasing. They are getting very threatened, only subsisting in some protected areas. But also the problem is that when people go and log those species, they also destroy the ecosystem around them sometimes. So the impact is not only on the species that is extracted, but also on the whole ecosystem. And there are many ways where DNA barcoding could help tackle these problems. Of course, in isolation, it would not be the solution to all problems, but it can contribute to solve them. Because DNA barcoding basically helps you to identify species based on their DNA. And so, as I was just showing with rosewood, one way identifying species can be useful is to be able then to track what is exploited and what is commercialized, and to distinguish between products that are coming from protected areas that should not be logged, for instance, and products that are sustainably sourced. So one way DNA barcoding can support conservation is to help controlling the trade of species. But also, for instance, it can tell you what species you have in, I don't know, your beauty cream or your food supplement. And like this, we can certify products as sustainably sourced or not. So that's when you test for adulteration. Another way knowing the identity of a species can be useful is if you want to detect invasive species at early stages. So instead of having to wait that the plant grows to be able to identify it. Maybe you can take a piece of soil and try to identify the seeds that are inside this soil by looking at their DNA. And like this, you can take decisions to protect the ecosystem in an early stage. Another way, being able to identify species where that can support conservation is simply by finding new species or species new to science, because if you don't name those species, then they cannot be assessed for their conservation status, so they cannot be part of conservation programs. So that's also a simple way where using DNA can help you find species that are different from any other known species, genetically speaking. And finally, because the DNA barcode is a piece of DNA, you can actually use the DNA barcodes to make, to elucidate relationships between your species, like producing phylogenies. And this can also support conservation. For instance, if you're interested in protecting high phylogenetic diversity or protecting species that are particularly distinct from a phylogenetic perspective. So all these applications of identifying species based on their DNA can contribute to biodiversity conservation. And there are many ways to identify species based on their DNA, and I will go quickly through the main ways. And of course, you will have to look for more resources to have a complete view, but I hope at least that will give you an introduction. So, as I said, the principle, the idea of DNA barcoding is that by looking at the DNA of a plant or a spider, an insect, or a fungus, you can figure out what species this is. And the first people to have brought this concept forward is Paul Ebert and his team. So that was 20 years ago, and they demonstrated that it was possible on animals. And so here I put just a little figure to illustrate the idea, which is that you have species that you know. So for instance, here, species A and species B. And if you look in their DNA, you can find regions that we will call DNA barcodes. So those are short regions in which you have a difference between species A and species B. So those are regions that can distinguish between both species. And then the idea is that if you have an unknown sample, like species X here, you can sequence the same region of the DNA, and then by comparing it with the barcodes of species A and species B, you can figure out what species X is. So once they demonstrated that on animals, plant people really wanted to do the same. But also it was actually quite easy to find a good DNA barcode region for animals in the mitochondrial genome. It was not that easy for plants. It took them like 10 years to find something satisfying. Because the mitochondrial genomes of plants is evolving differently from animals, and it's not very it evolves more slowly, so it's not very helpful to distinguish closely related species. And at the same time, it varies a lot structurally, so it can be difficult to amplify sequences from it. So then, because of that, people looked not in the mitochondrial genome, but in the chloroplast genome of plants. So that's where they actually found what we call often the traditional plant barcodes. So they tested a few different regions in the chloroplast genome, and they settled on RBCL and MATK as like the main barcodes, but you will see that people also use others. And notably, another one that is sometimes used is one that is in the nuclear genome of plants, which is the ribosomal internal transcribed spacer, so ITS we call it. And an important characteristic of those traditional barcodes is that they are present in many copies in one plant cell, because the chloroplast genome is repeated in the cell, and because the ITS region is repeated in the nuclear genome. And this is important because it makes them a bit easier to sequence, even when you don't have much DNA at hand. So those are like the traditional barcodes, but unfortunately, even those were not perfect for plants. It's still quite difficult in some groups to distinguish species just based on those regions. So people have been looking for more regions that could be used as plant DNA barcodes. And what has helped this search in the last maybe 5 years is that sequencing DNA has become cheaper. So now we can do high throughput DNA sequencing, which provides a lot of data in which we can look for new regions that can be efficient as barcodes. So I'll just go very quickly through 3 main high throughput DNA sequencing approaches that people have used to find barcodes. So the first one is the obvious one, is simply to sequence the complete genome of the species you're interested in. That's great because when you do that, you get everything. So you get the full plastome, mitochondrial, and nuclear genomes of your plants, in which you can find that you can then compare between species to identify good barcodes. And because you get everything, you also get the traditional barcodes. So you can still use them if you want. But the problem with that approach is that it's very expensive. So not everyone can do that at all, actually. So people have been looking for cheaper methods, and one of them is what we call genome skimming. So it's again sequencing the genome, but you don't invest so much in the sequencing. So at the end, you will not get the full genome. You will just get a skim of it, so a random section of it. And because it's random, the regions that are repeated in the genome are more likely to be sequenced. And if you remember, I was telling you that the traditional barcodes are repeated in the genome. So actually, by using this method, you do get the traditional barcodes. And in addition, you get also more regions that tend to be repeated. So typically what you get with this method is a full chloroplast genome that then you can compare between species, either to identify smaller regions in it that can serve as barcode, or you can also use the full chloroplast genome as a barcode that has been proposed by some people. And it can work very well for some species. But sometimes it's not yet enough. For instance, I work on palms, and palms, their chloroplast genome doesn't evolve super fast, so it can be difficult to distinguish closely related palm species or populations just based on the chloroplast genome. And then another and last technique you can use is what we call target capture sequencing. So in that case, you will not sequence randomly, but you will sequence regions that you know and that you want to target. So for instance, a few hundreds genes from the nuclear or the plastic genome, whatever. So you will use some methods to fish those regions from the genome and to sequence them preferentially. And so it's great because it's cheaper than sequencing the whole genome, and it's a bit more consistent, less random than just doing genome skimming. And the other great thing about it is because it's not perfect, it also does, it also catches some fragments randomly. And again, because it's random, then repeated regions have more chances to be caught randomly. And so you often actually end up also with getting the traditional barcodes when you use this region. So this is to illustrate that using traditional barcodes is not incompatible with using new barcodes and high throughput DNA sequencing. So the high throughput sequencing just allows you to try more barcodes if you have specific cases in which the traditional ones don't work. So this is to find small regions that can be used as a barcode. But there is another approach to DNA barcoding, which instead of using small regions, just uses the whole genome as a kind of barcode. So the idea here is that instead of targeting a few regions, you will look at the whole composition of the genome, and then you will compare the composition of the genome between the species, as you would do for when you compare sequences. So how do you measure the composition of the genome? Well, you can use what we call the K-Mer approach. So it sounds a bit weird, but K-Mer is simply just a stretch of K nucleotides. So for instance, in my example here, K equals 5. And so we are looking at 5 mers, which are just sequences of 5 nucleotides. And the idea here is that so you sequence your genome with one of the approaches that I mentioned before. Typically will be the genome skimming approach. So you have like a little random fraction of your genome. And then in that fraction, you check all the, like you check all the K-Mers patterns that you have. So if you're looking at 5 mers, then you check all the sequences of 5 nucleotides that you find in that genome. And for each pattern, so for instance, you have the ATGCT pattern. For each pattern, you count how many times this pattern occurs in your genome. And you do that for every pattern and for every species, and that gives you profiles of K-Mers for each species that you can then compare, and assuming that they are characteristic enough of the species so that they can serve as a genome-wide barcode. So you can find more details in one of the papers that presents this method, for instance, that I put here. So that's how you design your barcodes. But then how do you actually identify your species based on the barcode? I'll just spend a couple of minutes explaining this now. So the principle is very simple. Here it's a bit of jargon, but the idea is to match a query against a reference data set. So in our case, the query is the DNA of the species we want to identify, and the reference data set comprises the DNA barcodes of all the species that could be the species that we want to identify. Okay, so if I work on palms and I have an unknown palm, then I need a reference data set with all the palm species. So that's, and then I will compare the DNA of my sample to the DNA in the reference. And if I find a close match, then I might have solved my problem and identified my species. So for instance, here in the example on the top, we can see that the sequence of species X, which is the one to identify, is exactly the same as the one of species A, whereas it's much more distant from the sequence of species B. So the conclusion from that is that species X is probably species A. But of course, this will only work if you have all the species in your reference. Because imagine you have a sample of species C that you're trying to identify. If you don't have C in your reference, you're never going to be able to say, you're never going to be able to find a match. And you might find a close match to A, but the match to C would be closer, and you would have no way to know that. So the completeness of the reference is very important in the DNA barcoding process. And there are like 3 methods to actually do the matching of the query against the reference. So very briefly, the most simple one is to look at similarity. So that's the example I was showing. And then the other example is to look at the similarity, but of composition instead of the similarity of just the nucleotides. So that's the example I was describing with the K-Mers. In both cases, you can calculate genetic distances and then hope that your closest match is actually the right species. And another approach, which is a bit different, is the phylogenetic approach. Instead of just looking at similarity, you will model the evolution of DNA through time. So by doing the phylogeny with the typical phylogenetics approach, like maximum likelihood phylogenetic inference and things like that. And then you will get a tree. And for instance, here, species X falls closer to species A than to species B in the tree. And that can give us an indication that species X is in fact species A. So those are the three main methods, and there are many softwares that allow to implement them. So I encourage you, if you're interested, to look into this. But I don't have time to go more into the details, but I hope you understood the principles. And now I want to go a bit, I want to be a bit less technical and look more at the potential of this approach to solve real-world problems. So I will illustrate that with cases where DNA barcoding has been applied already. So going from research to impact does require to tackle technical and societal challenges. So a case where people have already been from research to impact is the case of Cycad trade monitoring. So Cycads are plants that are highly praised by collectors. And so there is a lot of illegal trade going around, and it has a big impact on Cycad's populations. It's driving some species to extinction, notably in South Africa. And so the South African researchers and government have really decided to tackle that. So this has started as research because they first had to look at what could be a good barcode for cycads, and so they identified regions that would work well to distinguish cycad species or even potentially populations, because some populations are in protected areas, whereas others are less protected. And then they use this information to find out what species were traded on local markets for medicinal remedies, but also what species were exported or traded by collectors, because cycads can be sold for thousands of pounds. So those are two different uses of cycads, and obviously one is more about having a sustainable livelihood, like trying to live out of the natural resources, while the other case is really trying to make money, regardless the impact it has on the species. So it's very important to know that we are not talking about necessarily the same problems, and they might not be solved using the same incentives or enforcement approaches. But in both cases, DNA barcoding allowed the people in South Africa to know what cycads were traded, and in some cases to prosecute people for illegal cycad trade or poaching. So that's a nice example, and it has required political will and research and a lot of work. But besides the societal challenges, there are also a few technical challenges that first need to be solved when you want to apply DNA barcoding, and I'm going to go through a few of them now. So the first one that I mentioned a bit already is to really have a comprehensive, up-to-date, reliable reference data set. as I explained earlier. And so one example that we are familiar with at Q is the example of rattans. So rattans are palms from mostly Southeast Asia, which are used to make furniture by local people, but also by international companies such as IKEA. And in most cases, rattans have to be collected from the wild. And so far, we don't really know how that impacts the wild populations. We don't know even what species are really in the furniture. So we are now collaborating with IKEA to try to find this out. So first, to find this out, you have to be able to get the DNA out of furniture, but you also have to be able to match this DNA against the comprehensive reference data set of all 450 rattan species. But because we have the luck of having a lot of well-identified collections of rattans and to be able to collaborate with taxonomic experts of this group, then we are in a good trajectory to actually have a complete reference data set for this group that comprises multiple samples per species, and for which every species in the reference has a voucher in a herbarium, so that we can then check their identity if necessary. And the taxonomy of the group has been recently solved, and this was the condition for this project to succeed. Without that taxonomical revision, we could not make that project, because how can you identify a species if you don't even know species boundaries in your group? So the taxonomy here, as in many cases, is again a fundamental basis of DNA barcoding. But so there are other challenges, and one of these is to find the right barcode. So a barcode, an ideal barcode, has to vary between species, but be more conserved inside species. So that's what people talk about sometimes as the barcoding gap, which I represented like in a schematic way here on the little graph on the right. So this is to illustrate a study that has been done by Brazilian researchers who designed a DNA barcoding project to find out what was in acai products. So was it the true acai, which I think is Euterpe oleracea, or was it something else, for instance, Euterpe edulis? And to do that, you have to be able to distinguish Euterpe edulis and Euterpe oleracea. And so you need to have like a bigger genetic variation between the species than inside the species. And so they managed to find a region that was working for them and that allowed them to show that some acai products are adulterated with something else. So I think it's a nice example. And then another challenge of DNA barcoding is when you have more than one species in a sample. Like there are some samples for which it's easy. Like if I try to identify a rattan cane, then I know it's just one species. But if I have a curry powder or a soup and I want to try to find out what is in there, then I have to be able to separate the DNA of those species in the lab and or during the analysis of the data. So this is a whole other set of work and of analysis that people refer to as DNA meta barcoding, and it's very useful for many applications like authenticating food products or monitoring, water quality, soil quality, etc. And I think that's my last challenge. So another challenge of DNA barcoding is that sometimes the DNA is degraded. So typically in processed food products, for instance, but also typically in timber products. You can have high DNA degradation which means that sometimes the traditional barcodes are too big compared to the size of the DNA that you get. And so then you have to use expensive techniques, or you can design mini barcodes, which are shorter and therefore can still work with degraded DNA. And so this is a big challenge in applying DNA barcoding to monitoring timber trade. But people have already applied DNA barcoding with success to monitor if trees were coming from protected areas or non-protected areas, or if timber products or logs were from protected species or non-protected species, etc. So here again, there is a lot of development going on and a lot of potential. And many private companies are already using this technique to develop certifications based on DNA identification. And it's a good thing because it can help consumers choosing between projects that are certified as sustainable or not. But of course, it's also very important that we make sure that DNA barcoding is or becomes accessible to all. And there are many aspects to accessibility. So of course, it's important to make sure you can carry your lab in the rainforest, for instance, and sequence on site, etc., or in places that maybe have a less reliable access to electricity or internet. So there is this aspect of accessibility that has to be solved, and there are many developments on that too, with mini sequencing machines or mini labs. But it's also important that things like this stay cheap and that they're adapted for each application. And this can only be figured out by designing your DNA barcoding project with the end use in mind. And I think maybe most importantly, to improve accessibility of DNA barcoding, the analytical tools, the reference databases, have to be accessible to everyone. And this is a vision of the International Barcode of Life project, whom they are really putting traditional barcodes, sequences at least, available to everyone in their database. And of course, there can be in the future more databases with more barcodes or even full genomes shared across everyone on Earth. And it's the same for analytical tools. Like there are some really nice open source softwares that are available to actually perform the DNA barcoding once you have the data. But it's not as easy as just putting everything online. There is also considerations to have in relation to who accesses those data, and not everyone can access data even if they are online, and also how do we share the benefits from DNA barcoding so that it's fair. And I don't necessarily have the answers to all of that, but I just wanted to put these in your mind too, and that's how I will finish actually. So to conclude, I hope I managed to show you that DNA barcoding is not just one thing. It covers many methods and barcodes, and it can support plant conservation, and it already does so. But that requires to tackle technical and socioeconomic challenges in relation to the reference data set, the informativeness of your barcodes, the practicality of the tools, but also, it requires general public and political will, and to consider matters such as benefit sharing or how do we protect plants and still ensure livelihoods. So yeah, I'm done, and I have no idea of the time or if anyone is listening.
Yes, we are listening you. So thanks a lot for it's I have a couple of questions.
Is it possible to see you instead of seeing my screen?
Well, I'm seeing you. I'm not so sure how to configure it. Are you still
Don't worry.
sharing?
This is too complicated.
Sorry, you can just maybe not sharing anymore, so maybe we could see.
Oh yeah.
So at the beginning, so it's amazing how is the potential, and we think about neotropical region, right? Because when we think about tropical regions, let's say very bio-rich in terms of species and with many species, unknown species.
Yeah.
And you ended the talk referring to the importance of good taxonomic work and also the reference library. But how can we discover new species, for instance? Because I think we are a bit far from have a good reference library for tropical regions, right? For biodiversity regions, hotspots, and we are losing species every day. When you talk about DNA barcode of wood, I just remember about the trucks of wood going out of the Amazon every day. Do you think there is somehow to solve this about discovering new species, and how can we manage without the good reference library? How?
Yeah. So actually, that's for discovering or naming new species. You don't necessarily need a good reference. You just need to have characterized what you know, and then you can go to the forest and then sequence everything that is around you. And if you find something that is different from anything that you have in your reference, then you will think, oh, maybe that's something new. And then you can look at this in more detail. And people have done that for insects, and like they have studies where they took this Minion sequencer to I think Borneo, and they just went there and they found like 600 new species of flies. Of course, you also have to define what you mean by a species. That's another discussion.
Yeah, that's another point. Okay.
But yes, you can definitely.
And when the last time you really updated me about the DNA barcode. I didn't have any clue about this, detecting patterns of this
The K-Mer, yeah.
little things. And I think that is very promising, no? Because considering that the traditional, the problems regarding the traditional DNA barcoding, I really thought that it could be very promising method.
It can be, but it's very sensitive to how much you sequence, obviously, because it's just you taking a random fraction of your sample, of the genome of your sample, and you still need to take enough so that consistently you get always the same profile for the same species, you see. So it might not be the cheapest approach, and in many cases, just going for a nice little piece of DNA might just be what you want.
Yeah, no, I get the problem. It really can be expensive, right? That's the main problem.
Yeah. If you check the paper, you will see they show the minimum amount of coverage of the genome you will need to have some reliability. I think it's promising in some cases, but sometimes it might just be an overkill.
Okay. So we have one question here. When there is more than one species in a sample, how do you separate the sequence that did not correspond to a species from the bank? I mean, it should be the reference library.
Yeah, so you can do different things. Like you can extract your DNA, and then you could do some cloning and sequence physically your samples, and then physically separate your samples, and then sequence each fragment separately, either with traditional sequencing or with next generation sequencing technologies, which go fragment by fragment. And then when you analyze the data, you can see, you can separate them because they are different. Yeah. And so then you can match that to the reference database. But I'm not sure if I answered well the question or not.
No, I think so, yeah. So, thanks a lot, Sidonie, for
Thank you for inviting me.
your contribution to our event. And those who didn't, who is still not those who didn't do the inscription in the event, please go there. We still have time and are open for the presential event. And see you next talk.
Bye. Thank you.
Bye. So.